Hello viewers, welcome once again to our ongoing series on genetics entitled Mendelism and Beyond. In about half a dozen of the lectures previously interacted with the fundamentals of Mendelism, the departures from Mendelian principles, the linkage and our present scenario on sex determination. So in my previous lecture, I had discussed the environmental theory of sex determination, wherein we highlighted the role of hormones, temperature, nutrition, age, and even infection by parasites to show how these factors would affect the sex ratio of various species. Actually, the bipotential gonads in these organisms, they gave zygotes which perhaps had the capability to switch on and off the genes for a particular sex at the cost of another. And what would determine this? This was obviously determined by the external environmental conditions. So that means, again, to highlight what we discussed on the last turn, the diagram on your screens again signifies that in some cases it is the genotypic factors. The genotypic factors would outweigh the environmental ones and there will be a normal sex determination, whereas in many of them it would be the environmental factors which would prevail and would result even in sex reversal in the organisms during their lifetime. Let us today discuss one of the very interesting genotypic sex determining systems, and this would be the sex determining mechanism in Drosophila. As you are already aware, viewers, we have discussed the genotypic sex determination at length, and we saw that there are various systems, namely XXXY, XXXO, O here signifies that there is no chromosome either X or Y, and ZOZZ and ZWZZ type of mechanisms. The point is that Drosophila has the same general chromosome composition as humans. That is, it has the autosomes plus the XX and autosomes plus the XY. So we can safely assume that it is a member of XXXY type of sex determination, and therefore it is obvious for us to think that in Drosophila too, perhaps maleness is due to the Y chromosome. Let us see in our future course of discussion that this assumption is perhaps totally erroneous. Because if we have a normal case of XXXY, as you can see on your screens, then you have the simple XX, which is the female gametes, and XY, which are the male gametes. And in this case, the true progeny, the males and the females, would be got in a proportion of 1:1. Now, this would signify that in an XX and XY system, the females are homogametic, the males are heterogametic. And in our earlier discussions, we had classified this system as the ligase type of system in which it was first discovered. And of course, human beings along with other mammals and Drosophila, they all come into this category. Talking specifically of the fruit fly Drosophila melanogaster, viewers, you should remember that its diploid number is 8, and there are, out of these 8, or let us say 4 pairs, there are 3 pairs of autosomes and 1 pair of sex chromosomes X and Y. The females are X and X and males are XY. Now this description reads exactly the same as we find in our cells, except for the difference of the number of autosomes. But then the question arises that XO in case of Drosophila is a male. Imagine, it has no Y chromosome and yet it is a male. And more interestingly, XXY is a female. So that means in the presence of a Y, still the sex is being determined as a female. Now how does it happen? Because we are used to seeing the human genetics. In human beings, for example, XO will refer to monosomy because there is one chromosome less, and this results in an abnormality which is called as Turner syndrome. And you know, viewers, that individuals with Turner syndrome are short statured, they are sterile because the ovaries they fail to develop, and they have lots and lots of heart problems and other medical and developmental ailments. And on the other hand, XXY is a case of having 3 chromosomes, that means 1 additional chromosomes, and this in humans results in Klinefelter syndrome, and because there is an extra copy of the sex chromosome, and this will also lead to many physical and physiological aberrations. We will have a detailed account of all these chromosomal aberrations once we are amidst the study of human sex determination and human genetics. So that means before understanding as to how did this happen, how XO was a male and how XXY in Drosophila is a female, let us look at the screens to find out the morphology of the chromosomes of male and the female Drosophila. The naming of the chromosomes in the karyotype is also different from our conventional Denver system of the classification of the chromosomes, because we always talk about the chromosomes being arranged in a descending order. Here the sex chromosomes have been given priority and they constitute the chromosome pair number 1. As you can see on the screens, the pair number 2 and 3 are there, but the 4th pair is just like a dot. That means the number of genes would be few in that case. And the Y chromosome in case of Drosophila is virtually J-shaped. A scientist by the name Patterson was the first person to have discovered that the Y chromosome is in fact heterochromatic and it doesn't play any significant role in sex determination. You remember, viewers, when we were talking about linkage, we distinguished between complete linkage and incomplete linkage, and there we had discussed that the Y chromosome in Drosophila was an example of complete linkage and all the genes were being transmitted together. So according to Patterson, the first view which came was that the ratio between the X chromosome and the autosomes would perhaps govern the development, whether it is a male or a female. He talked something about the dosage. He said that if there is a single dose of X chromosome in a diploid, it would turn a male, and if there was a double dose of the X chromosomes, it would turn into a female. Now these were the initial ideas, but then the actual experimental proof came through the famous work of Calvin Blackman Bridges, whom we normally call CB Bridges, who in the year 1921 he crossed a triploid. Now viewers, you should remember a triploid is designated as 3N because it has 3 copies of each chromosome, and this was a female with a normal diploid male, that means it was XY, and he observed so many different combinations of the autosomes and the sex chromosomes. How would this XXX, say for example, arise? Actually, there is a non-disjunction between the X chromosomes during meiosis, and this would lead to the formation of triploids, because one of the pole would have 3, the other one would have 1. So non-disjunction we can define as the failure of a paired chromosomes to segregate during the first or the second anaphase of meiosis, and obviously it would produce 2 types of abnormal gametes. As you can see on your screens, one of the gametes would be having 1 extra chromosome. We will designate it as n plus 1. The other one would lack a chromosome because it has gone to the first one, so it will be n minus 1. And obviously, if the gametes are such aberrant gametes are going to fertilize, they would give rise to 2n plus 1 or 2n minus 1, depending upon which gamete is fusing with which one. On your screens, viewers, you find a cross performed by C.B. Bridges, where we have a triploid 3a plus XXX and a diploid male parent with 2a plus XY. And the kind of gametes which we find are 2a plus XX, and in the other case a plus XX and 2a plus X. So these are the various combinations which are possible. More interesting aspect is to see that we have a triploid female. If you are a little careful, you will try to interpret the ideas. A triploid female is 3 autosomes and 3 X chromosomes. Things seem to be a bit equal. Whereas the 3a plus XXX happens to be a triploid intersex, diploid male, triploid intersex, super female which is a meta female, and a diploid female. Why did we get these different types of combinations? Calvin was the one who suggested that sex in Drosophila is determined by actually a balance between the ratio of autosomal alleles. Now, autosomal alleles are favoring maleness, and the alleles which are present on the X chromosomes, they are favoring femaleness. Calvin didn't talk anything about why, because he said it was a balance between the autosomes and the X chromosomes. So this type of sex determination is called as the genic balance system or the gene balance theory. Now, this particular theory we will find on your screens, you will find that there is a sex index which has to be followed, which means it is a mathematical expression to find out what is the number of X chromosomes in relation to the autosomal sets. Not the number of autosomes, autosomal sets. So that means autosomal sets can be diploid, triploid or tetraploid, and we designate it as X by A. Now, on your screens you will find a type of a balance. If there are more of XX, naturally you can expect a female progeny, and if it is the opposite, you have more of autosomes, then the tendency is more towards maleness. Now, on your screens you would find again different types of combinations which are there along with the matings of the triploid female and a normal diploid male. And you can find various ranges which are there as far as the X by A ratio is concerned. Please pay attention, right from the top it happens to be 1.5 and then it is 1. Where it is 1, then it is a female, and again 1, it is a female. But then if the proportion becomes more than 1, then it becomes a super female or a meta female. Let us go down. If it is 0.75, which is of course it is less than 1, it is an intersex. If it is 0.67, it is still an intersex. But the moment this ratio reaches 0.50, that means it is something like X by ratio is 1 by 2, then it becomes a male. The ratio again is a male in 0.5. The moment the ratio is below 0.5, then it becomes a super male or a meta male. So this chart gives us various lessons to follow. So that means XXY flies are normal and XO flies are sterile females. So where does Y come in this picture? Presence of Y in the XXY did not cause any maleness, and its absence in XO flies did not produce any femaleness. So this is the answer to our earlier question that we had asked ourselves as to how does it happen, because Y has no role as far as the determination of maleness is concerned. But then why is Y there? We will just see in a minute. The normal fly is either XX or XY, and there are 3 chromosomes, and X chromosomes is going to determine the femaleness. As I said before, the fundamental point is ratio of X chromosomes to the autosomes. Here is a summary of the gene balance theory on your screens, which suggests that if X by A is greater than 1 or 1, then you can think of a female or a meta female. If it was greater than 1, it was a meta female. If the ratio is 0.5 but less than 1, then it is intersex. If it is 0.5 or less, then it is male. Now here is the answer to our question, that why is Y present there? Y chromosome in fact controls male fertility. So that means if Y is there, then, and if it is a male, that is XY, in that case it would be a fertile male. So that means Y chromosome controls male fertility per se and not maleness itself. So that means we find that if we compare this with the human chromosomal complements, we have 22 autosomal pairs, and the set in Drosophila has it has autosomal set, which we call in our ratio X by A, is to be taken the autosomal pair of each of the 3 chromosomes. So this has led to a situation to classify the genotypic sex determination mechanism even into 2 categories, because we always thought that it is a simple XXXY. No. There is one situation among the genotypic sex determination mechanism where the Y chromosome is responsible for determining sex of an individual, because it is the one which confers maleness into the progeny. So this is one. And the one which we are discussing right now is the X chromosome autosome balance system, which we have seen in Drosophila. Viewers, you remember, we also talked about Caenorhabditis elegans. There also the ratio between the number of X chromosomes and the number of set of autosomes was responsible for the determination of sex. Here, Y chromosome is required for male fertility. It doesn't determine sex. So that is the difference between the 2 mechanisms which we classify under the genetically controlled sex determining mechanisms. There is another very interesting aspect which is called as gynandromorphs in Drosophila. In fact, the existence of gynandromorphs also supports the idea of gene balance theory. If you just break up the word gynandromorphs, it means gyne and andro. That means female and male morphology at the same time, which means one part of the organism would have male characters and the other one would have female characters. Actually, these gynandromorphs would arise due to misdivision of the chromosomes. There would be a loss of X chromosome during development, and again this is an ample proof that Y has no role in sex determination here. When the zygote starts with a female, it has a normal 2A plus 2X. Something goes wrong. One of the X chromosome is lost during cell division. So if it is lost, then one daughter cell would have 2A plus 2X. The other one would be deficient in 1. And it will give rise to 2A plus X, that means 1 less. And the blastomeres with unequal number of chromosomes will give a bilateral gynandromorph. What do we mean by a bilateral gynandromorph? As you can see on your screens, the misdivision has taken place. One of the X chromosome is lost because it did not reach any pole. It was midway and it got lost. And the diagram on the right shows that within the same fly, one of the parts, half, is female and the other one is a male. There's another classical example of white and red eye in a bilateral gynandromorph. As you can see on the screen, viewers, the gene for the eye color is lost at the time of anaphase, and therefore one of the gametes receives only XO, that is 1, and the other one receives XX, and one of the gene is lost. So within the same Drosophila, one of the eyes is red and the other one is white. You must have read, viewers, that red eye color is dominant over white eye color. This we would be studying in details in sex-linked inheritance or X-linked inheritance. So that means the gynandromorphs are going to exhibit different proportions, and sometimes it may happen that there are many different cell lines with different chromosome number within the same organism. In this case, we would call them as chimeras or sex mosaics. Like you have a mosaic flooring, you have a random distribution of different types of chips. Likewise, it will be male, female in a random manner, the cells which are distributed in the body of an organism. Just to sum up the whole discussion, let us talk at the end about the genes which are involved in the determination. And now various genes have recently been identified. One of them is a sex lethal, which is SXL. It plays a critical role because it is the master switch for sex determination. This master regulator gene will control cell division and cell differentiation. Mind you, this gene is X-linked and it is present in both males and females. Activation of this gene is absolutely necessary for female development, provided the XA ratio is 1. You remember we said if the XA ratio is 1, then it will be a normal fertile female. Genes on the X chromosome which are present and they regulate the XSL into the state of female development, they are called as numerator genes because we have the X by A. So they are the numerator elements because they act on the numerator of the X by A of the gene balance situation. On the contrary, genes are which are present on the autosomes because we had the X by A and they act to regulate the SXL into off state. Off state would be the male development stage. They are called as the denominator elements. If we were to look at the diagram on your screens, you would find how to read this X by A ratio. If the numerator is 1 and the denominator is 2, of course the X by A signal ratio is 0.5. Now the gene SXL is off. If the situation comes where X and A are in the same number, then the signal ratio is 1, which means now the SXL is on and this is going to be a female. So that means it is the ratio of the SXL which controls another gene, which is again an autosomal gene. It is called as a transfer gene. Imagine, viewers, this gene had been discovered 80 years earlier by Alfred H. Sturtevant, one of the most illustrious students of Professor T. H. Morgan, who also gave the Morgan units. Now, the females which are homozygous for TRA gene are transformed into sterile males. And there are two different types of genes of TRA1 and TRA2. So they are present in both males and females. TRA2 is absent in males. The SXL gene controls another gene which is called as the double sex gene, abbreviated as DSX. This particular gene is present in both males and females, and it suppresses the female character. Just to summarize, in a female, the DSX gene is going to be activated so as to give rise to a female phenotype, and in the male, it will be the DSXM which will be activated, and it would give rise to the male differentiation genes. There is another gene which is called as the intersex gene. It is terminal in position and it will bring about female sex differentiation only along with the DSXF. And finally, a fruitless gene which is needed for courtship in males. So that means we find that depending upon how the initial RNA transcript of the DSX is processed, there are different proteins which are formed, and then they activate either the male or the female specific genes which are required for sexual differentiation. I may add a very important point here, that each step in this regulatory cascade would require gene splicing. And as you can see on your screens, the gene splicing of SXL, which is transcribed only in the females, is depicted, and so is the SXL of protein regulation of alternative splicing of its own mRNA. So as a result, we would find that the X by A ratio actually regulates the transcription of the SXL gene, which is the master gene itself. Various mutations can also affect the two sexes differently, wherein you will have now the different types of phenotypes of XY and XX respectively. So in other words, to summarize, we find, as on your screens you will find, that the XN and XY have different modes of genes which are activated or suppressed at different times. We will conclude by saying that although Drosophila has been categorized along with human beings as far as its XX, XY sex determination is concerned, there are many differences. For this discussion, I have selected the bibliography of Snoosted and Simmons, Russell, Klug and Cummings et al. And Pierce. I would also advise you to go through some of these books because they are the standard treatises. Thank you.